Hormesis is not a wellness concept. It is a dose-dependent biological law describing how living systems respond to stress. Every adaptive improvement in human physiology is triggered by controlled disruption followed by repair. Muscle growth, cardiovascular efficiency, metabolic flexibility, immune competence, and even aspects of cognitive performance all depend on this cycle. Without stress, there is no signal for adaptation. Without recovery, there is no construction of adaptation. Biology only improves through repeated cycles of challenge and restoration. Hormesis is a dose-dependent biological response in which low to moderate exposure to a stressor activates adaptive cellular repair pathways that increase resilience, while excessive or chronic exposure overwhelms these systems, suppresses recovery, and accelerates functional decline and biological aging. This definition matters because it explains why identical stressors can be either beneficial or harmful depending on dose, frequency, and recovery time. At the cellular level, hormetic stress activates highly conserved survival pathways. These include EMPK signaling, transient mTOR inhibition, increased NAD+ utilization, heat shock protein expression, and controlled production of reactive oxygen species. These signals are not damage in themselves. They are triggers that instruct the cell to shift from growth mode into repair and maintenance mode. Two of the most important repair processes activated under hormetic stress are autophagy and mitophagy. Autophagy is a cellular recycling system in which damaged proteins, dysfunctional cellular components, and metabolic waste are broken down and repurposed for energy or rebuilding. It is essential for maintaining cellular quality control. Without autophagy, cellular debris accumulates, increasing dysfunction and disease risk over time. Mitophagy is a specialized form of autophagy targeting mitochondria. Mitochondria are the energy-producing organelles of the cell, and they are highly susceptible to damage from oxidative stress and metabolic overload. Through mitophagy, dysfunctional mitochondria are selectively removed and replaced with new, more efficient mitochondria. This process is critical for metabolic health, endurance, and aging biology. Hormetic stressors such as exercise, fasting, caloric restriction, and thermal exposure can activate both autophagy and mitophagy. However, this only leads to improved function if recovery conditions allow rebuilding. Otherwise, activation of these pathways becomes chronic degradation rather than renewal. The key distinction in biology is not whether stress activates these pathways, but whether the organism has the capacity to complete the cycle. Activation, breakdown, repair, supercompensation. Stress itself is not harmful. Chronic unresolved stress is. In acute conditions, stress activates the sympathetic nervous system and the hypothalamic pituitary adrenal axis. Cortisol rises to mobilize energy, glucose availability increases, and attention narrows. This state is evolutionarily conserved because it increases survival in short-term challenges. However, cortisol is not designed to remain elevated indefinitely. It is a pulsed signal, not a constant state. When stress becomes chronic, through sleep deprivation, emotional strain, excessive training load, poor nutrition, or circadian disruption, the same pathways that support adaptation begin to impair function. Persistent cortisol elevation disrupts insulin signaling, reduces tissue sensitivity to anabolic signals, impairs immune regulation, and contributes to systemic inflammation. One of the most critical variables in determining whether stress becomes adaptive or destructive is sleep. Sleep is not passive rest. It is a biologically active recovery state that regulates endocrine balance, immune recalibration, neural network reorganization, and metabolic restoration. During deep non-REM sleep, growth hormone secretion increases, tissue repair accelerates, and energy systems are restored. REM sleep contributes to emotional processing and synaptic reorganization in the brain. Sleep is also tightly linked to autophagy regulation and metabolic cleanup processes. Chronic sleep restriction reduces the efficiency of cellular repair systems, impairs glucose metabolism, and increases inflammatory signaling. Even when diet and exercise are optimal, insufficient sleep prevents full recovery and therefore limits hormetic benefit. Training stress follows the same principle. Resistance training and high-intensity exercise induce controlled micro-damage in muscle tissue, deplete glycogen stores, and generate metabolic stress signals. These signals activate anabolic repair pathways, leading to muscle hypertrophy, improved mitochondrial density, and increased neuromuscular efficiency. However, if training load exceeds recovery capacity, the system does not adapt upward. It degrades. Performance declines, injury risk increases, and systemic stress markers remain chronically elevated. Fasting operates through similar mechanisms. Energy restriction activates AMPK, inhibits mTOR, and promotes autophagy and mitophagy. This can improve metabolic flexibility and cellular maintenance. But fasting combined with poor sleep, high psychological stress, or excessive training can shift from beneficial signaling into energy deficit strain. Caffeine introduces another layer of stress modulation. It blocks adenosine receptors, increasing alertness and stimulating catecholamine release. This can enhance performance and cognition, but it also masks fatigue signals. If caffeine is used to override insufficient recovery, it increases total allostatic load rather than improving resilience. Allostatic load is the cumulative physiological burden imposed by chronic stress exposure without adequate recovery. It represents the net cost of maintaining stability under repeated or prolonged stressors. When allostatic load remains low, the organism cycles efficiently between stress and recovery states. When it remains high, physiological systems begin to lose flexibility and aging accelerates. The central principle is not stress avoidance. Stress avoidance is biologically incompatible with adaptation. The principle is stress calibration. A properly functioning biological system exhibits three properties. It tolerates stress, it adapts to stress, and it returns to baseline efficiently. Failure in any of these three produces dysfunction. If stress is too low, there is no adaptation stimulus. If stress is too high, repair capacity is overwhelmed. If recovery is insufficient, even moderate stress becomes cumulative damage. Indicators of healthy hormetic balance include stable or improving sleep quality, consistent energy levels, normal or improving resting heart rate, stable or increasing heart rate variability, preserved cognitive function, and progressive performance improvements over time. Indicators of maladaptive stress accumulation include persistent fatigue, worsening sleep, reduced heart rate variability, elevated resting heart rate, decreased motivation, and declining performance despite continued effort. Biological systems do not respond to intention, discipline, or motivation. They respond only to net physiological load over time and the adequacy of recovery mechanisms. Longevity, therefore, is not achieved by minimizing stress. It is achieved by maintaining a controlled oscillation between stress and recovery in which adaptive signaling is strong enough to stimulate repair pathways such as autophagy and mitophagy, but recovery capacity is sufficient to complete the rebuilding phase without chronic physiological strain. In this framework, resilience is defined not by resistance to stress, but by the speed and completeness of recovery after stress exposure.